Okay, so endomembrane system. Endomembrane system is a collection of organelles who share some jobs. So one job that the endomembrane system organelles and structures share is that they're responsible for protein traffic. They're responsible for the production, not all the production, but they're responsible for the packaging and the movement of proteins within the cell. The other function that they have is that they're very tied to the metabolic functions within the cell. So the day-to-day living, the chemical reactions that allow this cell to perform its functions. Endomembrane systems have the following components. Okay, a nuclear envelope, the endoplasmic reticulum. We'll talk about rough versus smooth in a couple of slides. Golgi apparatus, the lysosomes, the vacuoles, and the plasma membrane. So the thing about the endomembrane system, or how we know who belongs in the endomembrane system, is these guys are all either directly connected, meaning that the membranes are physically attached to one another, or they're joined together by vesicles, meaning that we can transmit or transport proteins or something else from one of these to another using a vesicle. Okay, we'll see that when we talk about moving stuff from the endoplasmic reticulum to the Golgi, or even what the lysosomes and vacuoles do. So, endoplasmic reticulum, let's start there. I want you to think about the endoplasmic reticulum as a factory that's specifically responsible for synthesis, for building things, building biological things that the cell needs. Structurally, it makes up more than half of the total membrane in most of the eukaryotic cells. The endoplasmic reticulum is continuous with the nuclear envelope. So remember, we had that double lipid bilayer. Coming off the external portion of that double lipid bilayer, we're going to move right into the endoplasmic reticulum, and specifically the rough endoplasmic reticulum. And if we keep following that membrane out, it's going to directly connect in with the smooth endoplasmic reticulum. The difference between rough ER and smooth ER? Ribosomes. So if you're rough endoplasmic reticulum, you're rough, you're bumpy, and you have ribosomes stuck to your surface. If you're not, if you're a smooth ER or SER, then you don't have ribosomes stuck to your surface. Artist rendition on top, transmission electron micrograph below, rough endoplasmic reticulum with all the ribosomes stuck to it. As we move further out, you'll notice you have those membrane folds, but you no longer have the little bumps attached. Okay, so functions. Smooth endoplasmic reticulum functions are going to be different than rough endoplasmic reticulum functions. They have different structures, and we keep saying this. Structure equals function, function equals structure, right? So these are the big four things that the smooth endoplasmic reticulum is responsible for. These are the big four I want you to remember and hang on to. You're going to come back and you're going to need these again. Even if we don't specifically use them, and we do, but if we don't use them in depth here in 181, you're going to need them when you hit Biology 201 and even 202. You need to understand these and commit these to memory. So the things that the smooth ER is responsible for is the synthesis of lipids. So things like steroids and cholesterol. We'll talk about that later. We're going to metabolize carbohydrates. We're going to detoxify poisons, and we're going to store calcium. So those are the big four that the smooth endoplasmic reticulum is responsible for. The rough endoplasmic reticulum, again, different structure, different function. So remember, it has the ribosomes that are bound to it. And what those ribosomes on the rough ER do is they secrete glycoproteins. Look at that word, glyco and protein. Well, protein's obvious. Break it apart. Protein's obvious. I got a protein. Now look at glyco. I look at glyco, I see glyco, glycogen, glucose. I see sugar. And when I see sugar, I think of the macromolecule carbohydrate. So what we have here is we have proteins that are covalently bound to carbohydrates. What the rough ER does is twofold. What it's going to do is it's going to distribute transport vesicles. So it's going to have little protein, or not even necessarily so little, but it's going to have proteins that get surrounded by membranes into those little transport vesicles that we talked about, and then it's going to send those transport vesicles out away from the surface of the endoplasmic reticulum to other parts of the cell. One of the big stops for the transport vesicles from the rough ER is to the Golgi, and we'll talk about that. The other thing that the rough ER does is it's the membrane factory for the cell. So if somebody needs a membrane patch, if we need a new membrane for a new organelle, it's the responsibility of the rough endoplasmic reticulum to go ahead and make that membrane. Golgi apparatus. So the Golgi apparatus is made up of flattened membrane stacks. You'll hear a lot of people refer to it as like a pancake, a stack of pancakes back and forth. It has those stacks. Sorry, should have said that while I was here. Those stacks or those folds, those flattened things are called cisterna. Plural is cisternae. Okay. The job of the Golgi apparatus is like a pack and ship. So I think of Golgi apparatus as pack and ship, and I'm discovering that more and more of my students don't really comprehend a pack and ship anymore. So you can also kind of think of it like an Amazon warehouse. Most of us can relate to Amazon warehouse. They don't make anything. We don't make anything in the Amazon warehouse. We modify some stuff. We break up packages of 10 into, singles and doubles that get shipped out. So we're going to modify stuff. We're going to repackage stuff, so we're going to put, so let's say I order from 3 different vendors on Amazon and it all comes in one box. Well, that's because somebody in the warehouse pulled this part and this part and this part together and put them into a package for me. The difference in the Golgi is that it manufactures certain macromolecules, and that's really difficult for us to talk about at this level, but the Golgi has the ability to take smaller subunits and put them together, almost like Legos. So it's not going to manufacture them from nothing, but it's going to put monomers together into a polymer form. Okay. So 3 big functions of the Golgi. It's going to modify products that came from the ER, from the endoplasmic reticulum that got there via vesicle. It's going to chunk things together into macromolecule format, and then it's going to sort and package materials into different transport vesicles, kind of like what happens in an Amazon warehouse. Oops, sorry about that, wrong direction. There's your image. It has a cis side and a trans side. So the cis side is the receiving side. It's where vesicles are going to come in. They're going to fuse with this membrane. Contents are going to get dumped into the actual Golgi cisternae so that they can be rearranged, repackaged, maybe 2 molecules chunked together into a macromolecule, and then they're going to work their way through the Golgi and exit on the trans face, the shipping side, as whatever vesicle needs to go. Some of those vesicles will stay within the cell and have a function there. Others will migrate to the plasma membrane and be exported out of the cell. So here's a good stopping point for us. You want to be able to compare and contrast the rough endoplasmic reticulum, that's your RER, rough endoplasmic reticulum, your smooth endoplasmic reticulum, and your Golgi apparatus. So remember, in a compare and contrast, you have things that are similar and things that are different. So your Venn diagrams are a great way to accomplish that. They're certainly not required, but they're a great way to help you organize your thoughts. So when you talk about compare and contrast, you want similarities and contrasting or differences. So I'm going to pause here and we will pick up with lysosomes.